Assalamualaikum and good day everyone. So today we're going to learn about chapter 6.3, which is protein synthesis. So in protein synthesis there are two stages, which is transcription and translation. So we're going to learn about transcription first. So there are three stages in transcription, which is initiation, elongation and termination. So first we're going to learn about initiation first. The first stage is initiation. So this is an example of DNA, which consists a lot of genes, but so this is a gene which codes certain protein, and this is the RNA polymerase or ribosomal polymerase. It will slide to DNA to find promoter, and it will attach to the promoter and make the DNA unwind or uncoil. Moving on to the next stage, which is elongation. So RNA polymerase will synthesize the RNA nucleotide from 5' to 3' by adding RNA nucleotide which is complementary with the template. And so RNA polymerase will synthesize the RNA nucleotide from 5' to 3' until the termination site. Last stage is termination. So RNA polymerase will detach from the termination site, and mRNA will move away from DNA to produce mRNA here. Okay, so after RNA polymerase detach from termination site, the DNA will rejoining back. Okay, and the mRNA here will call it as a pre-mRNA because it consists intron and exon, and this pre-mRNA will proceed for next process. We are going to learn about translation. So translation happens after transcription and DNA replication, and translation occur in cytoplasm. There are three stages in translation, which is initiation, elongation and termination. But before we learn about initiation, we have to know and learn about amino acid activation. Amino acid activation means activate the amino acid by binding the amino acid to tRNA. So this is the tRNA. Okay, so this is the tRNA. So the amino acid will bind to at the 3' of the tRNA here. Okay, the amino acid will bind at the 3' of the tRNA. So as we can see here, this UAC is an anticodon sequence where anticodon will complementary with codon on mRNA. Okay, so every tRNA has anticodon that codes for certain amino acid. So for this UAC will complementary with AUG, and this AUG will code for amino acid methionine here. So methionine will bind to on the 3' on tRNA. Okay, with the help of amino acyl tRNA synthetase. This enzyme will help methionine amino acid to bind on the 3' of the tRNA, and this structure is called amino acyl tRNA. Okay, so amino acyl tRNA is this structure is called amino acyl tRNA, like this. Okay, so so small ribosomal, okay, this is a small ribosomal subunit will come to bind the mRNA and tRNA. Okay, and the binding will form small ribosomal subunit, and mRNA and tRNA. Okay, and so this is the initiation process. So to complete the initiation process, large ribosomal subunit, large ribosomal subunit will come to bind with the complex. So amino acid here, this amino acid which attached to the mRNA will occupy P site on large ribosomal subunit. Okay, so this is the complete initiation complex. We go to the second stage of translation, which is elongation. So there are three main steps in elongation, which is codon recognition, peptide bond formation, and translocation. So we go back to this structure here. So incoming. So the incoming. This is the incoming amino acid tRNA will enter to a site of ribosome, which anticodon on tRNA must be complementary with the codon on mRNA. So GAU is complementary to CUA. So for the second step is peptide bond formation. So between two amino acid, peptide bond will form. Peptide bond will form. So this is a peptide bond. Peptide bond will form between two amino acids, and this process is catalyzed with the enzyme peptidyl transferase. And then the third step is translocation. So the ribosome will move a codon ahead, which means it will be like this. So the tRNA on A side will move to P side, tRNA on P side will move to E side. The tRNA on E side will move out. So this process will repeat with the next incoming amino acyl tRNA, which will enter to the A site, and peptide bond will form between two amino acid, and which will catalyzed by peptidyl transferase enzyme, and the two ribosome will translocate again, move a codon ahead, and it will repeat until it reach to stop codon. Go to the final stage of translation, which is termination. So when A site, here we go back to this diagram. So when A site of ribosome reaches stop codon, which consists UGA, it could be UGA, UAA, or UCA, but here is UGA, so stop codon. A protein called release factor will bind to the A site, to the stop codon. Usually the tRNA, right? But this time, since here is UGA, stop codon, so the protein called release factor will bind to the A site, to the stop codon. So this release factor causes the addition of water. And when the molecules of water will enter the complex and hydrolyze the bonds, and this polypeptide will be released from the complex. So and besides that, water also hydrolyze the bonds between small and large ribosomes, and the bonds between mRNA and tRNA. So the endocytoplasm is left with the polypeptide. So this polypeptide is the product of the translation, which where we produce polypeptide chain, which is protein. Okay, thank you.